Hello there, and welcome to Simple Sleepy Science. Tonight, we're exploring the molecule of life itself, DNA, the code written inside every cell of your body. Right now, you're carrying about 6 feet of DNA in each of your 37 trillion cells. If you unraveled all of it and laid it end to end, it would stretch from here to the sun and back over 600 times. You're holding the distance to stars inside you, coiled so small you can't see it. This is your space for calm wonder, a place where science becomes a gentle friend as you settle into rest. So, get comfortable, let your breath slow, and let's explore the quiet miracles written in your genetic code. We start with something you've been doing since before you were born. If this brings you calm, if you enjoy these quiet journeys through science, please hit that like button and subscribe so you don't miss tomorrow's story. And leave a comment telling me where you're watching from and what time it is there. Obnakad of knowing you're out there, sharing this wonder with me. DNA looks like a twisted ladder. Scientists call it a double helix. 2 long ribbons wind around each other, connected by rungs like steps you could climb if you were small enough. But this ladder is impossibly small. You could fit 200,000 DNA strands across a single period on a page. Something that tiny holds every instruction needed to build you. The ladder is made from just 4 chemical letters: A, T, G, and C. That's the entire alphabet of life. 4 letters creating every instruction your body needs. A always pairs with T. G always pairs with S. This rule never breaks. Each letter finds its perfect match across the ladder's rungs, like locks finding keys. These 4 letters repeat 3 billion times in your cells. 3 billion letters spelling out how to make your eyes, your heartbeat, your ability to think and remember. All written in combinations of A, T, G, and C. The code for brown eyes sits in one section. The instructions for how your heart beats sit in another. Everything about you, written in 4 letters. The twist isn't random. The ladder spins exactly once every 10 rungs, creating a rhythm that runs through every cell. This spiral protects the code inside. It also helps proteins find the right spots when they need to read your instructions. Right now, proteins are moving along your DNA like fingers reading Braille. They feel the bumps and grooves, understanding which letters come next. They're reading recipes for building proteins that become your skin, your muscles, the neurons firing in your brain as you hear these words. If you pulled one cell's DNA straight, it would stretch 6 feet. But it's packed so carefully, it fits in a space smaller than you can see. Your cells can find any instruction instantly among billions of letters. The organization is beyond anything humans have invented. This same twisted ladder exists in every living thing. Bacteria have it, trees have it, birds carry the same basic structure. We're all using the same design, modified over time, but never completely changed. Life found a pattern that works and kept it for billions of years. Your DNA is being copied right now too. Every time a cell divides, it duplicates all 3 billion letters. The machinery makes fewer than 1 mistake per billion letters. It's precision that makes our best technology look clumsy. If this brings you calm, if you enjoy these quiet journeys through science, hit that like button and subscribe to join fellow curious minds who find peace in understanding. Leave a comment telling me where you're watching from and what time it is there. I love knowing we're sharing this moment across the whole world. The atoms making up your DNA didn't start here on Earth. The atoms in your DNA were born in dying stars billions of years ago. Every carbon atom, every nitrogen, every bit of oxygen and phosphorus that makes up your genetic code came from stars that exploded long before Earth existed. Here's how it happened. Stars are fusion factories. They crush hydrogen atoms together to make helium. Then, helium fuses into carbon, carbon into oxygen. The process continues, building heavier and heavier elements. But this only happens in the cores of massive stars, where temperatures reach millions of degrees. When these giant stars run out of fuel, they explode. These explosions, called supernovas, scatter all those newly created atoms across space. Carbon, nitrogen, oxygen, phosphorus. The exact ingredients needed for DNA. These atoms travel through the cosmos for millions of years, drifting in clouds of gas and dust. Our solar system formed from one of these clouds about 4.6 billion years ago. As Earth took shape, it collected these stellar atoms. They landed in oceans, mixed with other elements, and eventually organized themselves into the first simple molecules, then more complex ones, and finally, DNA. The phosphorus in your DNA's backbone came from a star. The nitrogen in your genetic letters was forged in stellar fire. You're not just connected to the cosmos, you're made from it. Your body is recycled stardust that learned to think and feel and wonder about where it came from. Every person who ever lived carries these same stellar atoms. Your DNA and Einstein's DNA contain carbon from the same ancient supernovas. The nitrogen in your genes might have floated through space alongside the oxygen, now in a tree outside your window. We're all built from the same cosmic recycling program. This matters because DNA is 3.5 billion years old as an idea. The first DNA appeared on Earth when our planet was still young and violent. That original molecule found a way to copy itself and pass information forward through time. Every living thing today descends from that first successful code. Your DNA isn't just your instruction manual. It's a message passed down through an unbroken chain of life stretching back billions of years. Every ancestor you had, going back through humans, through earlier primates, through ancient mammals, through fish, through the first single-celled organisms, all of them successfully copied their DNA and passed it forward. You're the current carrier of instructions that never stopped being copied since life began. The atoms might be ancient, forged in stars, but the pattern is even older. The way those atoms arrange themselves into the double helix, the 4-letter alphabet, the pairing rules. These patterns emerged billions of years ago and proved so effective that life never abandoned them. Every creature uses the same basic system because it works. Right now, you're lying there, a temporary arrangement of stardust, carrying a pattern older than mountains. Your DNA connects you not just to other humans, but to every living thing. The same code that tells your heart to beat also tells bacteria how to move and trees how to grow. We're all speaking the same molecular language, and that language is being read and copied in your cells right now, following rules written in the hearts of dying stars, carried through space, and assembled on this small planet into something that can contemplate its own origin. Inside your cells right now, molecules are reading your DNA, like musicians reading sheet music. But here's the interesting part. They don't play every note. They choose which sections to perform based on what your body needs at this exact moment. A heart cell reads the heart genes. It ignores everything else. A brain cell skips past the heart instructions and reads only brain genes. Your stomach cells are reading recipes for digestive enzymes, while your eye cells read instructions for detecting light. Same complete score in every cell, but each plays only its part. This is how one fertilized egg becomes an entire human with hundreds of different cell types. Every cell starts with identical DNA. As cells divide and specialize, they don't lose genes. They just silence most of them. A liver cell still carries the instructions for making eyeball proteins. It just never reads those pages. The conductor of this orchestra is chemical signals. Hormones tell certain genes to turn on. Proteins bind to DNA and say, "Read this section now." Your environment sends messages that change which genes your cells express. When you eat, digestive genes activate. When you exercise, muscle-building genes turn on. Your DNA responds constantly to what's happening around it and inside you. This system allows incredible complexity from simple instructions. You have about 20,000 genes, but your body makes over 100,000 different proteins because cells read genes in different combinations, skip sections, or read the same gene multiple ways. It's like having a cookbook where the same recipe produces different dishes depending on which steps you follow. Right now, your immune cells are reading genes that make antibodies. Your skin cells are reading instructions for making keratin, the protein that protects you. Your bone marrow is reading genes that produce new blood cells. All of this happening simultaneously, each cell knowing exactly which part of the genetic score to perform. And cells can change their performance. If you get infected, immune cells start reading different genes to fight the invaders. If you go outside on a sunny day, skin cells read genes that produce melanin for protection. Your DNA isn't static instructions. It's an interactive library that responds to your life. Temperature affects which genes turn on. Stress changes genetic expression. Even your thoughts and emotions send chemical signals that influence which genes your cells read. Your experiences literally change how your DNA is used, creating a constant conversation between your life and your code. This is why identical twins with identical DNA can look different as they age. They're reading the same genetic sheet music, but conducting different performances based on their unique lives. One twin might activate genes, the other keeps silent. Same notes, different symphonies. The most amazing part is the coordination. Your body has 37 trillion cells all reading DNA simultaneously, each performing its specific role, creating a harmony that keeps you alive. Brain cells fire, while heart cells pump, while stomach cells digest, while skin cells protect. All following the same basic score, but playing different parts. And none of this requires your conscious control. Right now, as you rest, your cells are conducting this molecular symphony without any input from your thinking mind. They know which genes to read, when to read them, and how long to keep them active. It's automation at a level we barely understand. This automatic system running in your cells uses the same basic instructions found in every living thing, which brings us to something that sounds absurd but reveals a beautiful truth. You share about 60% of your DNA with a banana, not because you're banana-like, but because all life on Earth descended from one ancient ancestor. That first spark of life, appearing in warm oceans roughly 3.5 billion years ago, carried the fundamental toolkit for living. Build cells, process energy, copy yourself, repair damage. Every living thing inherited these basic instructions. Bananas build cells almost the same way you do. They have DNA wrapped in a double helix. They use the same 4 letters, A, T, G, and C. Their cells read genetic instructions and make proteins following the same basic rules your cells use. At the molecular level, the process is nearly identical. The shared 60% covers the essentials: instructions for cell division, energy production, protein building, DNA repair. These functions are so fundamental that life never redesigned them. Evolution modified and improved the original toolkit, but kept the core mechanisms because they work perfectly. The 40% difference creates everything that makes you human instead of a banana. Your brain, your ability to move, your complex organs. But the foundation, the deep cellular machinery, remains the same across species. We're all running variations of the original software. You share even more DNA with other animals. About 85% with mice, roughly 90% with cats, nearly 99% with chimpanzees. The closer the evolutionary relationship, the more genetic code we share. Every percentage point of difference represents millions of years of separate evolution. But the similarities reveal our common origin. This gets even stranger when you look at specific genes. Humans and fruit flies share a gene called eyeless that's involved in eye development. Completely different eyes, different structures, different capabilities. But the master switch that says, build an eye here, is recognizably similar. Life reuses successful designs across wildly different species. Your DNA contains genes that also exist in yeast, in worms, in fish. Scientists can sometimes replace a broken human gene with the equivalent gene from another species, and it works well enough to study. The code is that universal, that compatible across millions of years of separate evolution. This matters because it means we're not separate from nature. We're part of it. The tree outside your window is a distant cousin, running similar genetic programs. The bacteria in your gut are relatives, using the same DNA alphabet. Birds, fish, insects, all family at the molecular level. Every living thing is an experiment using the same basic instruction set. Some experiments grew leaves, others grew wings. Some developed brains that could wonder about their own existence. But we're all variations on the same theme, all descendants of that first successful molecule that learned to copy itself billions of years ago. Right now, in your cells, and in banana cells, and in every cell of every creature on Earth, DNA is being read using the same fundamental process, the same pairing rules, the same double helix structure, the same molecular machinery. Life found a system that worked and spread it across the entire planet, modifying it into millions of different forms, but never abandoning the core design. You're connected to everything alive through this shared code, this molecular language spoken by every creature that ever existed. This shared code, running through all life, faces constant threats. Right now, as you're settling down for rest, your DNA is being damaged. It happens thousands of times each day. Sunlight creates errors. Stress causes breaks. Even normal chemical reactions inside cells harm the code. But your cells have repair crews that patrol constantly. They move along your DNA looking for problems. A broken strand, mismatched letters, damaged sections. When they find errors, they fix them with remarkable precision. They cut out the damaged part, read the opposite strand to see what should be there, and rebuild the section perfectly. Sleep is when this repair work intensifies. Your body shifts resources toward maintenance. During deep sleep, your cells prioritize fixing DNA over almost everything else. This is when you heal at the most fundamental level, letter by letter, base pair by base pair. The damage comes from everywhere. Ultraviolet light from the sun confuse two letters together where they shouldn't connect. Free radicals, which are unstable molecules produced during normal metabolism, attack DNA constantly. Even the process of copying DNA during cell division creates occasional errors. Your genetic code is under siege every moment you're alive. Without repair, you'd accumulate errors quickly. Within weeks, your cells would lose the ability to function. Within months, you'd die. But repair enzymes work so efficiently that most damage gets fixed before it causes problems. They catch about 99.9% of all errors. The repair process is elegant. Enzymes recognize when the DNA shape looks wrong. They feel along the double helix, checking that everything fits together properly. When they find a bulge or a gap or letters that don't match, they stop and call in the repair machinery. Different types of damage require different repair methods. Small chemical changes get fixed by one system. Broken strands get mended by another. Major damage that affects both strands requires the most complex repair process, where the cell uses information from a sister chromosome as a template to rebuild the damaged section. Sleep deprivation disrupts this process. Studies show that people who don't get enough sleep accumulate more DNA damage in their cells. The repair crews still work, but they don't have enough time to fix everything before new damage occurs. Over time, this adds up. Your brain cells particularly benefit from sleep-based DNA repair. Neurons don't divide often, so they need to maintain the same DNA for decades. During sleep, brain cells ramp up repair processes, ensuring your neurons stay healthy and functional. This is one reason sleep is so critical for long-term brain health. Right now, as you lie here, repair enzymes are moving through your genome. They're finding yesterday's sun damage and fixing it. They're correcting errors from stress hormones. They're rebuilding sections damaged by normal cellular activity. Your body is investing in maintenance, making sure the code that builds you stays accurate. This happens every night. While you're unconscious, molecular machines smaller than you can imagine are reading your genetic instructions. comparing them to what they should be, and making corrections. They work in darkness and silence, asking nothing from your conscious mind, just faithfully maintaining the code that makes you possible. By morning, your DNA will be cleaner, more accurate, better maintained. Sleep isn't just rest. It's reconstruction at the molecular scale, ensuring the instructions that build you remain clear and functional for another day. Those repair mechanisms maintain your DNA sequence, but there's another layer to your genetic code that changes constantly. It's called epigenetics, and it explains why identical twins with identical DNA gradually become different people. Twins start as perfect genetic copies. One fertilized egg splits into two, creating two embryos with exactly the same 3 billion letters of DNA. At birth, their genetic code is identical. But from that moment forward, something remarkable happens. Their DNA begins to diverge, not in the letters themselves, but in how those letters are read. Think of DNA as a book. Epigenetics is like highlighting certain passages while leaving others unmarked. The text stays the same, but which parts get emphasized changes based on experience. Chemical marks attach to DNA, sitting on top of the double helix, telling cells which genes to turn on and which to keep silent. These marks respond to everything. Diet adds chemical tags to certain genes. Stress creates different patterns. Exercise changes which genes are accessible. Even emotions and thoughts send signals that modify how DNA is read. Your experiences literally write themselves onto your genetic code. One twin might experience trauma that adds chemical marks to stress response genes, making them more sensitive to anxiety. The other twin, living a calmer life, doesn't develop those marks. Same DNA, different expression. One twin might exercise regularly, activating genes involved in muscle growth and metabolism. The other stays sedentary, and those genes remain quiet. The marks are called methyl groups and acetyl groups. They're small molecules that stick to DNA or to the proteins DNA wraps around. They don't change the genetic letters, but they change access. A methylated gene is like a book whose pages are glued shut. The information is still there, but cells can't read it. This system allows your fixed genetic code to respond flexibly to your environment. You can't change the DNA you inherited, but you can influence how it's used. The genes are the hardware. Epigenetics is the software that decides which programs to run. Studies on identical twins show this clearly. Young twins have very similar epigenetic patterns, but twins separated at birth and raised in different environments develop increasingly different patterns over time. By age 50, their epigenetic marks can be dramatically different, even though their underlying DNA remains identical. This means your DNA isn't destiny. You're participating in how your genes express themselves. The genetic hand you were dealt at birth is just the starting point. How you live influences which cards get played. Even more striking, some epigenetic marks can be passed to your children. If you experience severe stress, certain marks might transfer to the next generation, affecting how your children's stress genes function. The science is still emerging, but evidence suggests your experiences can echo forward genetically. Your cells are adding and removing these marks constantly. Right now, based on what you ate today, how you felt, what you did, your epigenome is shifting. Some genes are becoming more accessible, others are being silenced. Your DNA is having a conversation with your life, adjusting its expression to match your circumstances. This flexibility is why humans can adapt to such different environments. We have the same basic genes, but epigenetics lets those genes respond to living in the Arctic or the tropics, to modern cities or traditional villages, to stress or peace, creating different outcomes from the same instructions. Those epigenetic marks control how your genes are read. But here's something surprising. Only about 2% of your DNA actually codes for proteins. The other 98% does something else entirely, and for decades, scientists didn't know what. They called it junk DNA, evolutionary leftovers, genetic garbage accumulated over millions of years with no real purpose. Just filler taking up space between the important genes. But that label is turning out to be completely wrong. Much of this so-called junk acts as switches and dimmer controls. These sections don't code for proteins themselves, but they regulate when and how much protein the actual genes produce. They're like the stage crew in a theatre. You don't see them performing, but without them, the show doesn't happen. Some stretches of DNA act as landing pads for regulatory proteins. When certain proteins bind to these sections, they can turn on genes located far away on the chromosome. Other sections act as insulators, preventing genes from interfering with each other. Still others produce RNA molecules that don't become proteins, but instead regulate other genes. Your genome is less like a simple instruction manual and more like a complex control system where most of the space is dedicated to regulation, not raw instructions. The 2% that codes for proteins is just the visible outcome. The 98% determines when, where, and how much of each protein gets made. Some of this non-coding DNA is genuinely ancient, fossilized remnants of old genes that stopped working millions of years ago. Evolution doesn't clean up perfectly. When a gene becomes unnecessary, it doesn't disappear. It just accumulates mutations until it's unrecognizable. These dead genes sit in your genome like abandoned buildings, serving no function but taking up space. Other sections are even stranger. About 8% of your DNA is viral. Millions of years ago, viruses infected your ancestors, inserted their genetic code into human DNA, and got passed down through generations. Most of these viral sequences mutated over time and lost their ability to make new viruses. They're just passengers now, hitchhiking through human history in your genome. But some viral DNA became useful. One ancient viral gene helps form the placenta during pregnancy. Without it, human mothers couldn't nourish babies in the womb. Evolution repurposed an invader into an essential human function. Your ability to exist came partly from a virus that infected an ancestor millions of years ago. Large sections of your DNA remain completely mysterious. Scientists can read them, see the letters, but have no idea what they do. They might be doing something subtle we haven't detected yet. They might be evolutionary experiments waiting to become useful. Or they might genuinely be junk, neutral remnants that don't help or harm enough for evolution to remove them. The humbling part is this: we can sequence your entire genome. We can read all 3 billion letters. We can compare your DNA to thousands of other humans and find every difference. But we still don't understand what most of it does. Your genetic code holds secrets we're only beginning to grasp. Every few years, scientists discover new functions hidden in the non-coding regions. What looked like empty space turns out to contain instructions written in a language we're still learning to read. Your DNA is more complex, more layered, more sophisticated than the simple gene-to-protein model suggests. You're carrying mysteries inside every cell, coded in sections of DNA whose purposes remain unknown. Those mysteries sit in DNA that stays relatively stable throughout your life. But there's one part of your genetic code that's shrinking right now. At the ends of each chromosome are protective caps called telomeres, and they're getting shorter with every cell division. Think of telomeres like the plastic tips on shoelaces. They don't contain any genes or important instructions. They just protect the real DNA from fraying. Each time a cell divides and copies its DNA, the copying machinery can't quite reach the very end of the chromosome. A small section gets left off. This isn't a mistake. It's built into how DNA copying works. The enzymes that duplicate your genetic code need to grab onto the DNA strand to start copying, and they can't grip the absolute end. So, with each division, chromosomes get slightly shorter. Telomeres take the hit. They're made of the same letters as regular DNA, just repeated over and over in a meaningless sequence. In humans, it's T to agua repeated thousands of times. These repeats act as a buffer zone. When cells divide, telomeres shorten instead of important genes getting cut off. Young cells have long telomeres, maybe 8,000 to 10,000 repeats. With each division, they lose 50 to 200 repeats. This happens throughout your life. By the time you're old, many of your cells have telomeres that are half their original length. Eventually, telomeres become too short. When they reach a critical length, cells stop dividing. They enter a state called senescence, where they're alive but frozen, unable to reproduce. This is aging at the cellular level, a countdown timer built into your chromosomes. Some cells never run out of telomeres because they have a special enzyme called telomerase. This enzyme rebuilds telomeres, adding back the repeats that get lost during division. Your stem cells have it, which is why they can keep dividing throughout your life to replace damaged tissue. Reproductive cells have it too, ensuring babies start with fresh, long telomeres instead of inheriting shortened ones. Most of your regular cells don't have active telomerase. Their telomeres just keep shortening until division stops. This prevents cells from dividing forever, which is actually protective. Unlimited division is cancer's trick. Cancer cells reactivate telomerase. That's part of how they become immortal, dividing endlessly when they should stop. About 90% of cancers have found ways to rebuild their telomeres, escaping the normal countdown that limits cell division. Scientists have tried extending life by activating telomerase in normal cells. In mice, it works. They live longer, stay healthier, but the cancer risk increases too. Telomere shortening isn't just aging, it's a safeguard against runaway cell growth. Your lifestyle affects how fast telomeres shorten. Chronic stress accelerates the loss. Poor sleep speeds it up. Smoking damages telomeres faster. But exercise seems to slow the shortening. So does meditation. A good diet helps preserve telomere length. Your choices influence your cellular clock. Right now, in cells dividing throughout your body, telomeres are getting shorter. In your gut lining, which replaces itself every few days. In your skin, constantly renewing. In your blood, making new cells to replace old ones. Each division ticks the clock forward. This is part of being alive. Your cells are counting their divisions, measuring their age in telomere length, following a program written into the structure of your chromosomes. You're a biological clock, keeping time at the molecular level. That clock ticks inside chromosomes that are far from static. DNA doesn't sit still like a blueprint stored in a drawer. It moves. The double helix breathes, twists, writhes, constantly shifting shape in response to what's happening in your cells. Proteins grab onto DNA and bend it. They loop it, twist it, pull distant sections close together. A gene at the beginning of a chromosome can be brought next to a gene near the end through this looping. Sections that look far apart when you draw DNA as a straight line are actually neighbors in the three-dimensional space inside your nucleus. This movement is essential for how genes work. A regulatory region might be 1 million letters away from the gene it controls, but proteins fold the DNA until those sections touch. The regulator can then directly influence whether the gene turns on or off. Your genome is constantly reorganizing itself, creating temporary structures that allow genes to communicate. The double helix also spins on its axis. As proteins read DNA or copy it, they create tension that makes the helix twist tighter in some places and unwind in other spots. Enzymes have to constantly manage this tension, cutting the DNA, letting it rotate to release the twist, then resealing it. Your genetic code is being cut and repaired thousands of times each day just to manage the mechanical stress of being read. DNA also vibrates. At the atomic level, the bonds holding it together are constantly flexing and bending. These tiny movements help proteins recognize specific sequences. The shape of DNA changes subtly depending on which letters are present, creating a landscape of bumps and grooves that proteins can feel as they move along the molecule. When genes turn on, the DNA in those regions becomes less tightly packed. The double helix loosens, allowing proteins to access the genetic information. Active genes are found in open, accessible regions. Silent genes stay tightly coiled, wrapped around proteins so firmly that the cellular machinery can't reach them. Your nucleus is organized into territories. Chromosomes don't float randomly. Each occupies a specific region, and active genes tend to cluster together in certain areas called transcription factories. These are like busy workplaces where multiple genes are being read simultaneously, sharing the necessary protein machinery. This organization changes throughout the day. Some genes are more active in the morning, others turn on at night. The physical structure of your DNA shifts to accommodate these changes, moving active regions to accessible locations and tucking inactive genes away in dense, quiet areas. During cell division, DNA condenses dramatically. It packs down into the familiar X-shaped chromosomes you might have seen in textbooks. This is DNA at its most compact, organized for transport into daughter cells. After division completes, the chromosomes unfold again, returning to their working structure. Even in this relaxed state, DNA is incredibly organized. 6 feet of it fits into a nucleus you can't see with your naked eye. The folding is precise, allowing any gene to be found instantly among billions of letters. The organization is dynamic, constantly adjusting based on what your cells need. Right now, your DNA is moving, looping, twisting, reorganizing. It's alive in a way that static diagrams can never capture, responding to every signal in your cells, adjusting its shape to allow genes to speak or stay silent. This moving, shifting DNA contains passengers that have been along for the ride for millions of years. About 8% of your genome isn't human at all. It's viral, remnants of ancient infections that integrated into your ancestors' DNA and never left. Here's how it happened. Certain viruses, called retroviruses, work by inserting their genetic code into the host's DNA. HIV does this. Once inserted, the viral genes use your cellular machinery to make new viruses. Usually, this happens in regular body cells. When those cells die, the viral DNA disappears with them. But occasionally, a retrovirus infected a sperm cell or an egg cell. When that happened, the viral DNA got passed to the next generation. Every cell in the resulting offspring carried the viral code, including their own reproductive cells. The virus became hereditary, traveling forward through generations. Most of these ancient viral insertions happened millions of years ago. Over time, mutations corrupted the viral genes. They lost the ability to make new viruses. They became fossils, inactive sequences just sitting in the genome, no longer dangerous. Evolution neutralized them. You're carrying thousands of these viral remnants. They're scattered throughout your chromosomes, broken fragments of viruses that infected ancestors you share with all other humans. Some insertions are so old you share them with mice and dogs, meaning the original infection happened before those species split from our lineage. Scientists can read these viral fossils and date them. They can track when certain infections occurred, which species were affected, how the viral sequences changed over time. Your DNA is an archive of ancient plagues, a record of diseases that threatened your ancestors millions of years before humans existed. But here's where it gets interesting. Some of these viral genes became useful. One viral gene, called syncytin, helps form the placenta. It allows cells to fuse together, creating the interface where mother and baby exchange nutrients and oxygen. Without this viral gene, human pregnancy wouldn't work. Evolution took an invader and repurposed it. What was once a threat became an essential part of human reproduction. The virus that inserted this gene infected a mammal ancestor about 150 million years ago. Every placental mammal, from mice to elephants to humans, uses this viral gene. Other viral sequences might be regulating human genes. Some sit near important genes and appear to influence when those genes turn on. Scientists are still figuring out which viral remnants are doing something useful versus which are truly inactive junk. Your immune system learned to tolerate these viral sequences. It has to, since they're part of your genetic code. But occasionally, one reactivates partially. It doesn't make infectious viruses, but it can produce viral proteins that your immune system recognizes. This might contribute to certain autoimmune conditions where the body attacks itself. You're a genetic mosaic, part human, part viral, part bacterial, if you count the mitochondria in your cells, which are descendants of bacteria that merged with our ancestors billions of years ago. You're not a pure lineage. You're a collaboration between different forms of life, merged at the genetic level. Those viral sequences move when your DNA moves, get repaired when your DNA gets repaired, and get copied when your cells divide. They're woven into your genome now, inseparable from the human DNA around them. Permanent passengers on a journey that began with an infection and became something more. These viral passengers, along with all your human genes, face an extraordinary spatial challenge. 6 feet of DNA has to fit inside a nucleus that's about 1/100th the width of a human hair. The compression required is almost beyond comprehension. DNA solves this through layers of organization. First, it wraps around protein spools called histones. 8 histone proteins cluster together, and DNA winds around them twice, like thread around a bobbin. Each spool holds about 150 letters of genetic code. This creates structures that look like beads on a string when viewed under powerful microscopes. These bead strings then coil into thicker fibers. Those fibers loop and fold into even more compact structures. The folding continues through multiple levels until you get chromosomes dense enough to see during cell division. It's organization within organization, each layer increasing the compression. The math is staggering. A single chromosome, if stretched out, might be several centimeters long. Compressed into its cell division form, it's only a few micrometers. That's a compression ratio of about 10,000 to 1. Imagine taking a rope long enough to stretch across a city and folding it so precisely it fits in a marble. But compression alone isn't enough. Cells need instant access to any gene among billions of letters. The organization has to be smart, not just compact. And it is. The packing isn't random. Active genes stay in loosely packed regions where proteins can reach them easily. Inactive genes get buried in tightly packed areas. The proteins that manage this organization are working constantly. Enzymes add or remove chemical marks that tell histones how tightly to grip the DNA. Other proteins pull loops of DNA through ring-shaped complexes, creating specific 3-dimensional structures. Your genome is being actively sculpted every moment. When a cell needs to turn on a gene, it has to unpack that section of DNA. Proteins arrive and push the histones aside, unwinding the DNA until it's accessible. The gene gets read, then the section gets repacked. This unpacking and repacking happens thousands of times per second across your genome. Different cell types organize their DNA differently. A liver cell packs its DNA with liver genes accessible and brain genes buried. A brain cell does the opposite. Same DNA, different organization. The physical structure of your genome helps determine what kind of cell you have. The nucleus itself is organized into territories. Each chromosome occupies a specific region and doesn't wander. Genes that need to work together often end up physically close, even if they're on different chromosomes. The three-dimensional organization brings distant genetic elements into contact. During cell division, the packing intensifies dramatically. The already compressed DNA condenses further into the tight X shapes of chromosomes ready for transport. After division, when daughter cells have received their DNA, the chromosomes relax again. They unfold into working structures, still incredibly compressed, but accessible enough for normal cellular function. This compression system is so efficient that it makes our best data storage look primitive. Computer engineers study DNA packing to understand how to store information more densely. Nature solved this problem billions of years ago, creating a system that combines extreme compression with instant access, maintained by molecular machines operating at scales we can barely observe. Your cells perform this organizational miracle in every nucleus, managing complexity that spans from individual atoms to structures large enough to see under microscopes. This organizational system responds to more than just cellular signals. It responds to your experiences, and those responses can sometimes be passed to your children. Studies on trauma survivors show something unexpected and controversial. Extreme stress can create chemical marks on DNA that transfer to the next generation. The clearest evidence comes from Holocaust survivors. Their children and grandchildren show different stress hormone levels compared to people whose families weren't affected. They have altered responses to stress, measurably different patterns of anxiety and fear. The trauma their ancestors experienced left biological marks that carried forward. These aren't changes to the DNA letters themselves. The genetic sequence stays the same. But the epigenetic marks, those chemical tags that control which genes turn on, can transfer through sperm and egg cells. Your grandfather's experiences might have added marks to stress response genes that you inherited. Scientists first noticed this in animal studies. Mice exposed to chronic stress had offspring that were more anxious, even when raised in calm environments with no stress. The babies inherited behavioral changes without inheriting any obvious genetic mutations. Something beyond the DNA sequence was being passed down. In humans, the evidence is harder to prove, but compelling. Children of people who experienced famine show different metabolism patterns. Descendants of trauma survivors have altered stress responses. The experiences of previous generations appear to echo through DNA, not as memories, but as biological sensitivities. This makes evolutionary sense. If your ancestors survived a famine, passing down a metabolism that stores fat more efficiently might help their children survive similar conditions. If they endured constant threats, passing down a heightened stress response might improve survival. The body tries to prepare the next generation for the world their parents knew. But in modern life, these inherited sensitivities can cause problems. A stress response calibrated for surviving war doesn't serve you well in peacetime. Metabolism optimized for famine creates health issues with abundant food. Your DNA might be responding to threats your ancestors faced, but you don't. The mechanism involves methylation, those chemical marks that sit on DNA and control gene expression. Normally, most methylation gets erased when sperm and egg cells form, wiping the slate clean for the next generation. But some marks survive this erasure. They persist, carrying forward into the embryo. Not all trauma transfers this way. Most epigenetic marks from life experiences do get erased. The ones that survive tend to involve extreme, prolonged stress during critical developmental periods. Your everyday anxieties probably won't mark your DNA in ways that affect your children. And there's hope in this system. Just as trauma can leave marks, healing might too. Evidence suggests that positive experiences, therapy, and stress reduction can change epigenetic patterns. You're not locked into inherited trauma. The marks can be modified through how you live. Your DNA carries whispers from the past, biological memories of what your ancestors endured. But you're also writing new marks, creating patterns that might pass forward. The conversation between genes and experience flows through generations, each inheriting the echoes of those who came before, while adding their own. Right now, your experiences are creating chemical marks on your DNA. Some will fade when you die. Others might persist, influencing descendants you'll never meet, carrying forward a molecular memory of your life. Those marks control which genes turn on, but there's another layer of complexity. Your cells don't just decide whether to read a gene, they decide how to read it. A single gene can be read in multiple ways, creating different proteins from the same genetic instructions. This process is called alternative splicing. Think of genes as recipes written in sections. Your cells can include all the sections, skip some, rearrange the order, or use different starting and ending points. Same recipe, dozens of different outcomes. Here's how it works. Genes contain sections called exons that code for proteins, separated by sections called introns that don't. When a gene gets read, cells first copy the entire thing, introns and all. Then, enzymes cut out the introns and splice the exons together. But which exons get included in the final message can vary. One gene might have 10 exons. Cells can use all 10, or skip exon 3, or use only exons 1, 4, and 7. Each combination produces a different protein. This is why humans, with only about 20,000 genes, can make over 100,000 different proteins. The math isn't one gene equals one protein. It's one gene equals many possible proteins. Your cells make these decisions based on what they need. A brain cell might splice a gene one way to produce a protein specialized for neurons. A muscle cell splices the same gene differently to produce a version optimized for muscle function. Same genetic information, different interpretations. Some genes are spliced in hundreds of ways. The gene that helps neurons communicate has over 38,000 possible splice variants. That's 38,000 different proteins from one gene. The complexity multiplies far beyond what the gene count suggests. Cells can also read genes backward. Some genes produce proteins when read in the normal direction, different proteins when read in reverse. The DNA sequence is the same, but reading it backward reveals different instructions, like how some words spelled backward create new words. Even the starting point matters. Genes don't always begin at the same place. Cells can start reading from different points in the sequence, producing shorter or longer versions of proteins. They can also stop at different points, again changing the final product. This flexibility allows evolution to work more efficiently. Instead of needing separate genes for every protein, life can store multiple instructions in the same genetic space. It's like having a compressed file that unpacks differently depending on which program opens it. Mistakes in splicing cause diseases. If cells splice a gene wrong, they produce a faulty protein. Some genetic disorders aren't caused by mutations in the gene itself, but by errors in how the gene gets spliced. The instructions are correct, but the reading process fails. Your cells are making these splicing decisions constantly. As proteins wear out and need replacement, genes get read and spliced based on cellular needs. The process is so complex that scientists are still discovering new splice variants for genes we've studied for decades. This means your DNA is even more information-dense than it appears. Those 3 billion letters don't just encode 20,000 genes. They encode a flexible library of instructions that can be read multiple ways, producing different outcomes depending on context, need, and cellular interpretation. Your genetic code is a language with grammar rules that allow endless creative variation. This creative variation within your genes creates the proteins that make you unique. But here's something remarkable. All human diversity, every difference in height, eye color, personality, and ability comes from just 0.1% of our DNA. The other 99.9% is identical across every human on Earth. That tiny fraction, just 3 million letters out of 3 billion, accounts for everything that makes individuals different. Your brown eyes versus someone's blue, your height versus theirs, your risk for certain diseases, your natural talents. All of it sits in that narrow slice of variation. The 99.9% sameness covers the fundamentals. The instructions for building cells, processing energy, making proteins, repairing DNA. Your heart works the same way as everyone else's heart. Your neurons fire using the same mechanisms. Your immune system follows the same basic playbook. The core machinery of being human is universal. Even that 0.1% difference isn't evenly distributed. Most of it sits in regions between genes or in regulatory sections. The actual protein coding genes are even more similar. When you compare the genes that make proteins, humans are nearly identical. The variation comes mostly from how those genes are controlled, not the genes themselves. This means racial categories have almost no genetic basis. The visible differences we use to sort people into groups represent a tiny fraction of tiny variation. Someone from one ancestry group might be genetically closer to someone from a completely different group than to someone who looks like them. Appearance is a poor guide to genetic similarity. Geographic ancestry does leave subtle genetic signatures. People whose ancestors lived in certain regions for many generations show patterns in that 0.1% variation. Scientists can sometimes identify where your ancestors lived by reading these patterns. But the similarities vastly outweigh the differences. 2 random humans share more DNA than 2 random chimpanzees from the same forest. Human genetic diversity is unusually low compared to most species. This suggests our entire species went through a population bottleneck sometime in the past, perhaps dropping to just a few thousand individuals. Everyone alive today descends from that small group, which is why we're so genetically similar. The differences that do exist are evolutionarily recent. Human populations only separated and spread across the globe in the last 100,000 years or so. That's barely any time in evolutionary terms. Not enough time for significant genetic divergence. We're one young species that hasn't had time to split into distinct groups. Your DNA proves connection, not separation. That person across the world who speaks a different language, practices different customs, looks nothing like you share 99.9% of your genetic code with them. You're running nearly identical software, following almost the same instructions to build a human body and brain. The 0.1% creates beautiful variety. Different combinations of these variable regions produce endless individual uniqueness. No two people, except identical twins, have exactly the same pattern. But this diversity rests on a foundation of overwhelming similarity. Every human is a slight remix of the same genetic recipe. The ingredients are identical. The proportions vary just slightly, and those slight variations create every personality, every face, every mind you've ever encountered. We're far more alike than different, right down to our molecular blueprints, carrying nearly the same instructions, expressed in subtly different ways that create the appearance of vast diversity from tiny genetic differences. Within that 99.9% similarity, some people carry variations that give them extraordinary DNA repair abilities. These aren't dramatic differences, just subtle changes in repair genes that make their cellular maintenance work slightly better. They age slower, they resist cancer more effectively, they bounce back from damage faster. Scientists study these people to understand why some bodies maintain their genetic code more successfully. It comes down to efficiency. Their repair enzymes work a bit faster or more accurately. Their cells catch errors earlier. Their systems prioritize maintenance more aggressively. The result is DNA that stays cleaner over time. One well-studied example involves people who live unusually long, healthy lives. Centenarians often carry variations in genes related to DNA repair and stress resistance. Their cells handle damage better than average. This isn't immortality, just a slower accumulation of the errors that drive aging. But here's the hopeful part. Genetics isn't the whole story. Lifestyle affects DNA repair significantly. You can't change the baseline you inherited, but you can optimize how well your repair systems function. Exercise boosts repair enzymes. Physical activity increases production of proteins that fix DNA damage. People who exercise regularly show less DNA damage in their cells compared to sedentary people of the same age. Movement literally protects your genetic code. Sleep gives your body time to focus on repair. During deep sleep, repair activity intensifies, fixing damage that accumulated during the day. Chronic sleep deprivation leads to measurable increases in DNA damage. Your repair crews need rest time to work effectively. Diet matters too. Certain nutrients support repair enzymes. Antioxidants reduce the damage that repair systems have to fix. People who eat more vegetables and less processed food show better DNA maintenance. You're feeding your repair machinery every time you eat. Stress harms DNA. Chronic stress generates chemicals that damage genetic material faster than repair systems can fix it. Stress also diverts cellular resources away from maintenance toward immediate survival responses. Your DNA accumulates errors when stress is constant. But stress reduction helps. Meditation has been shown to increase telomerase activity, the enzyme that protects chromosome ends. Even short meditation practices produce measurable effects on cellular aging markers. Calming your mind protects your genetic code. Smoking accelerates DNA damage dramatically. Tobacco smoke contains chemicals that directly attack DNA, creating more damage than repair systems can handle. Smokers accumulate genetic errors much faster, which is why smoking increases cancer risk. Your choices about what you inhale affect your DNA directly. Sunlight damages DNA. UV radiation creates specific types of errors that repair enzymes have to fix. People who protect their skin from excessive sun exposure show less DNA damage over time. Sunscreen isn't just cosmetic, it's genetic protection. Your DNA responds to how you live. It's a partnership between the genes you inherited and the choices you make. You can't change your genetic baseline, but you can activate protective mechanisms, optimize repair systems, and reduce the damage your cells have to fix. Right now, the lifestyle choices you made today are influencing how well your DNA gets maintained tonight. The exercise you did or didn't do, the food you ate, the sleep you're about to get. All of it affects how effectively your cells protect and repair the code that builds you, creating a daily conversation between your choices and your genes. Those repair systems maintaining your DNA carry information about where your ancestors lived. Scientists can read your genetic code and trace human migration patterns back through time. Every lineage, when followed far enough, converges on one place. Africa, roughly 200,000 years ago. Your DNA is a map. It shows the journey your ancestors took, leaving genetic breadcrumbs as populations split and moved across continents. Small mutations accumulate over generations. By comparing these mutations between people, scientists can reconstruct who descended from whom and when groups separated. Mitochondrial DNA provides the clearest trail. These are small circles of genetic material inside your cells, separate from the DNA in your nucleus. Mitochondria are the energy factories of cells, and they carry their own genes. The key detail is that mitochondria only pass through mothers. Your mitochondrial DNA came from your mother, who got it from her mother in an unbroken maternal line. When scientists compare mitochondrial DNA from people worldwide, all lineages point back to one woman who lived in Africa roughly 150,000 to 200,000 years ago. She's called mitochondrial Eve, though that name is misleading. She wasn't the only woman alive then, just our most recent common maternal ancestor through this particular genetic line. Y chromosomes tell the paternal story. Only males have Y chromosomes, passed from father to son. These also trace back to Africa, to one man scientists call Y chromosomal Adam. He lived somewhere between 200,000 and 300,000 years ago. He and mitochondrial Eve weren't a couple, never met, lived thousands of years apart. But everyone alive descends from their genetic lines. Nuclear DNA, the main genetic code in your cells, confirms this story. The most genetic diversity exists in African populations. This makes sense. because African lineages have been accumulating mutations for the longest time. Populations that left Africa carry subsets of African genetic variation, showing they descended from groups that migrated out. The migration pattern is clear in genetic data. Small groups left Africa around 70,000 years ago, probably through the Middle East. They spread along coastlines, reaching Asia and Australia. Some turned north into Europe. Others eventually crossed into the Americas. Your DNA shows which path your ancestors took. Different populations adapted to new environments. People in high latitudes developed lighter skin to produce vitamin D in low sunlight. Populations at high altitudes evolved better oxygen processing. Groups that relied on dairy farming developed lactose tolerance as adults. These adaptations happened quickly, within thousands of years, but their surface changes to the same underlying human design. Your genome carries markers of this journey, small variations that cluster by geographic ancestry, showing where your lineages spent time. Someone of European descent has genetic signatures from populations that lived in Europe for thousands of years. African descendants carry the deepest, most diverse genetic heritage, reflecting humanity's longest continuous history there. But we're all one species. The time since leaving Africa is too short for significant divergence. Humans never separated long enough to become truly distinct. We're one young species that spread across the planet recently in evolutionary terms, adapting to local conditions but remaining fundamentally connected. Your DNA proves you're African at the core. Everyone is. Those 3 billion letters in your cells tell a story that begins in Africa and branches out across the world, connecting you through unbroken lineages to ancestors who walked the same land, carried the same basic code, and passed it forward until it reached you. That ancestral DNA you inherited isn't quite identical throughout your body. The DNA in your skin cells isn't the same as the DNA in your liver. You're not one person genetically. You're a mosaic of slightly different versions of yourself. This happens because mutations occur constantly. Every time a cell divides and copies its DNA, there's a small chance of error. Most errors get caught and repaired, but some slip through. That cell now carries a mutation that its parent cell didn't have. When it divides, its descendants inherit that change. Over your lifetime, different cell populations accumulate different mutations. A patch of skin cells might share one mutation. A section of liver tissue might have another. Your body becomes a patchwork of cellular lineages, each with its own unique genetic additions to the code you started with. Most of these mutations are harmless. They occur in non-coding regions or change DNA letters in ways that don't affect protein function. Your cells keep working normally despite carrying slightly different genetic sequences. You don't notice because the changes are subtle and don't alter how cells behave. Some mutations happen early in development. If a mutation occurs in an embryo when you're just a ball of a few hundred cells, that mutation spreads to billions of descendants. You might have an entire organ system carrying a genetic variation that the rest of your body lacks. Scientists have found that skin is particularly mosaic. Every square centimeter of skin can have dozens of different mutations scattered across its cells. Most came from sun damage, UV radiation, creating errors that got passed to daughter cells. Your skin is a genetic patchwork map of sun exposure history. The brain is mosaic too. Neurons accumulate mutations throughout life, and since they rarely divide, each neuron keeps its unique changes. Your brain contains billions of cells, each carrying a slightly different version of your genome. This might contribute to brain function, creating diversity in how neurons behave. Cancer is an extreme version of this process. One cell accumulates multiple mutations that remove normal growth controls. It divides uncontrollably, creating a population of cells with their own mutated genome, separate from your normal cells. Cancer is your own cells evolving into a harmful variant. Blood cells show mosaicism clearly. Your bone marrow produces millions of new blood cells daily. Occasionally, a stem cell acquires a mutation that gives it a slight growth advantage. That cell's descendants multiply faster, gradually taking over more of your blood cell population. This is common in older people and usually harmless, but it increases cancer risk. Even twins aren't immune. Identical twins start with the same DNA, but from the moment the embryo splits, mutations begin accumulating independently. By adulthood, each twin is a unique mosaic. They share the baseline code, but have accumulated different changes throughout their bodies. You're plural on the inside. One body containing trillions of cells, grouped into populations, each population carrying subtle genetic variations. The you that started as a single fertilized egg has diversified into a community of related but not identical genetic lineages. This genetic diversity within you is mostly invisible. Cells function similarly enough that differences don't matter for day-to-day life. But at the DNA level, you're not a single genetic entity. You're a collection, a biological collaboration between billions of slightly different cellular versions of the same original code. This collection of slightly different genetic codes in your cells represents an astonishing amount of information. DNA is the most efficient data storage system known. 1 gram of DNA can theoretically hold 215 million gigabytes of information. That's more data than exists in most data centers. The storage density is almost impossible to comprehend. All the data humanity has ever created, every book, every movie, every website, could fit in a container the size of a shoebox if encoded in DNA. Nature solved data storage billions of years ago with a system that makes our best technology look primitive. Scientists have successfully stored digital information in synthetic DNA. They've encoded books, including entire encyclopedias. They've stored movies, converting video files into genetic sequences. One research team encoded an entire computer operating system, plus a film, into DNA and later retrieved it perfectly. The process works by converting digital ones and zeros into the 4 letters of DNA. A and C might represent 0, G and T might represent 1. Any digital information can be translated into sequences of these 4 letters, synthesized into actual DNA, and stored in tiny amounts of liquid. DNA storage has advantages beyond density. It's stable. DNA can last thousands of years without degrading if stored properly. Scientists have successfully sequenced DNA from fossils tens of thousands of years old. Compare that to hard drives, which fail after a few decades, or magnetic tapes that degrade in similar timeframes. DNA also doesn't require power. Hard drives need electricity to maintain data. DNA just sits there, holding information without any energy input. You can store it at room temperature or freeze it for even longer preservation. The information remains encoded in the molecular structure, waiting to be read. Your body is already using this system. Every cell carries complete instructions to build an entire human. That's roughly the equivalent of storing the information needed to construct something as complex as a skyscraper, including all wiring, plumbing, materials, and assembly instructions in something smaller than you can see. And cells can copy this information. Your body duplicates 3 billion letters of code every time a cell divides, with accuracy rates better than 1 error per billion letters. We can't copy digital data that accurately. Our error rates are orders of magnitude higher. The reading speed is remarkable too. Cells can access any gene among billions of letters almost instantly. They don't have to search through the entire genome sequentially. The organization allows direct access to needed information, like random access memory in computers, but far more efficient. Engineers are working on DNA-based data storage for archives. Information that needs to be kept for centuries, like historical records or scientific data, could be encoded in DNA. The technology isn't practical yet for everyday use, because writing and reading DNA is still slow and expensive. But for long-term storage, it's nearly ideal. Nature has been using this system since life began. Every living thing stores its operating instructions in DNA, copies them with stunning accuracy, and passes them forward through generations. Your cells are performing data storage operations that exceed anything humans have engineered, managing complexity at scales we're only beginning to approach with artificial systems. You're walking around with the most advanced information storage technology in the universe, carrying instructions refined over billions of years, compressed into spaces too small to see, yet accessible instantly when needed. This information storage system didn't appear fully formed. It evolved from simpler molecules over billions of years. But once DNA emerged, it never changed its basic structure. The DNA in your cells uses the same design as DNA in bacteria, trees, mushrooms, jellyfish. The same 4 letters, the same double helix, the same copying method. This uniformity isn't coincidence, it's inheritance. All life on Earth shares one origin, one moment when molecules first learned to copy themselves and pass information forward. That happened roughly 3.5 billion years ago, when Earth was young and violent. Nobody knows exactly how it started. The leading theory involves RNA, a molecule similar to DNA but simpler. RNA might have been the first genetic material, capable of both storing information and catalyzing chemical reactions. At some point, DNA took over the storage role because it's more stable, while RNA became the messenger between DNA and proteins. That transition happened before the common ancestor of all current life. By the time recognizable cells appeared, they were already using DNA as their instruction manual. Every living thing today descends from that original lineage, making all of us relatives at the deepest level. Your DNA is running software 3.5 billion years old. Not unchanged, obviously. Evolution has modified, refined, added to, and optimized the code billions of times. But the underlying system remains the same. 4 letters, base pairing rules, double helix structure, DNA polymerase enzymes that copy the code, all inherited from the first organism that got this combination right. This continuity is astounding. Your genetic code connects you in an unbroken chain to the first living thing. Every generation between then and now successfully copied its DNA and passed it forward. Not one break in the chain, not one failure to reproduce. An uninterrupted line of successful copying spanning billions of years and leading directly to you. The atoms in your DNA have been recycled countless times. Carbon atoms might have been part of a dinosaur's DNA 100 million years ago. Before that, they might have been in ancient bacteria. The atoms flow through life in an endless cycle, but the pattern, the information encoded in how those atoms arrange themselves, that's what gets preserved and passed forward. Some of your genes are recognizably similar to genes in bacteria. Not identical, but related enough that scientists can trace the connection. These are ancient genes, core functions that life needed from the beginning. Building proteins, processing energy, copying DNA. These fundamental operations have instructions that date back billions of years. Other genes are newer, appearing as evolution experimented with complexity. Genes for building brains are relatively recent, maybe 500 million years old. Genes for making antibodies are even newer, but they're all built using the same 4-letter alphabet, following the same chemical rules, stored in the same double helix structure that the first life used. You're a temporary arrangement of atoms, yes, but you're also the current expression of a pattern that never stopped being copied since life began. The information that builds you has been passed through an unbroken chain of successful reproduction, refined and modified over billions of years, but never losing its fundamental format. Your DNA isn't just yours, it's life's DNA, a shared heritage connecting you to every organism that ever existed, all using variations of the same basic code, all descended from one origin, all carrying forward the pattern that started billions of years ago in ancient oceans on a young planet. Everything comes together here, in this quiet moment, as you settle toward rest. You're carrying stardust, atoms forged in dying stars billions of years ago, organized into molecules that learned to copy themselves and pass information forward through time. Your DNA connects you to everything, to the first spark of life in ancient oceans, to every creature that ever lived, all using the same 4-letter alphabet, the same double helix structure, to ancestors stretching back through time, an unbroken chain of successful reproduction leading directly to you, to every human alive today, sharing 99.9% of your genetic code, despite looking different and living in different corners of the world. You're not separate from nature, you're nature examining itself, complexity and beauty and ancient wisdom wrapped into a form that can wonder about its own existence. The code in your cells is older than mountains, refined over billions of years, carrying instructions that built your body from a single cell and maintain it every moment you're alive. Right now, as you lie here, your DNA is working. Repair crews are moving through your genome, fixing damage from today, preparing you for tomorrow. Genes are turning on and off based on what your body needs. Cells are copying their DNA, passing instructions to daughter cells. The molecular machinery is operating without any conscious input from you, following patterns written billions of years ago. Your experiences today added chemical marks to your DNA, changing how certain genes will be read. The food you ate, the stress you felt or didn't feel, the movement you did or skipped. All of it influenced your genetic expression, creating a constant conversation between your life and your code. Tomorrow, this process continues. More repair, more copying, more adjustments based on what you experience. Your DNA responds to your choices, activating protective mechanisms when you treat yourself well, accumulating damage when you don't. It's a partnership between the genes you inherited and how you live. You're a temporary carrier of ancient instructions. The atoms will move on, recycled into new forms after you're gone. But the pattern, the information encoded in your DNA, pieces of it will pass forward if you have children. Or it returns to the pool of human genetic diversity, contributing to the species that continues after you. This isn't sad, it's connection. You're part of something vast and continuous, a story written in 4 letters that began 3.5 billion years ago and continues through you into the future. Life found a way to preserve information across deep time, and you're proof that it works. As you settle into sleep tonight, let this understanding bring peace. You're stardust that learned to think, ancient patterns that became conscious, instructions refined over billions of years and expressed temporarily as you. Complex and simple at once, connected to everything alive through shared molecular language. If tonight's journey brought you calm and wonder, hit that like button and subscribe. Tomorrow brings another quiet exploration of science designed for rest. Leave a comment about which DNA fact amazed you most. I love hearing from this community of curious minds, finding peace in understanding. For now, let your breathing slow. Let your body do its work, repairing and maintaining the code that makes you possible. You're being rebuilt molecule by molecule as you rest, following instructions older than you can imagine. Sleep well.